Hi, my name is Paula Olaya, and I will present the X-ray-free electron laser-based protein structure identifier workflow developed by Global Computing Lab at the University of Tennessee, Knoxville, in collaboration with Innovative Computing Lab and Riken, Japan. Let's start with an overview of the problem we are addressing. Proteins and other biological molecules are responsible for many vital cellular functions, and these functions are determined by the structure of the protein. There are many proteins with different structures, and even a single protein can exhibit several inner structures, also called as conformations. Therefore, identifying the protein structure and conformations can help solve problems such as determining cause of diseases and designing drugs. One way to determine the protein structure is by studying diffraction patterns that are generated by an X-ray-free electron laser beam applied to the proteins. These diffraction images can reveal inner protein structure. The goal of this project is to design, implement, and validate a framework for identifying protein structural properties, for example, orientation and conformation, from different diffraction patterns. Orientation refers to the placement of the incident beam with respect to the protein, and it's defined by two angles. Conformation are all possible states that a protein can have. We are working with the diffraction patterns of an elongation factor II protein. We present our solution, the X-ray-free electron laser-based Protein Structure Identifier workflow, or XPSI, which analyzes protein diffraction images and predicts the structural properties such as orientation and conformation. As you can observe in the diagram, our workflow consists of the input diffraction pattern images that are processed by an autoencoder. The autoencoder captures key information and produces a tensor representation of each pattern. The new latent space is used to train and validate traditional machine learning models such as k-nearest neighbors. We use a KNN angle regressor for predicting the orientation and a KNN classifier for predicting different protein conformations. Due to the amount of data and the need for an effective framework, high-performance resources are required for executing and identifying the proteins with XPSI. As you can see in the pink box, we execute our test on a single node Power 9 with 2 GPUs NVIDIA V100s. Another contribution of this poster is the measurement of the prediction accuracy and performance of XPSI. For the diffraction imaging datasets, including realistic noise obtained by having a low beam intensity, which creates noisier images. We define two tests. In the first test, we only predict orientation of a single conformation. In the second test, we predict orientation and type of conformation. I remind you that our dataset has two types of conformations for the same protein. From the autoencoder training performance, we observe that it takes double the time to process the images for test 2, meaning that the time to train and extract the new latent space is proportional to the input data. The same results are obtained from the training the KNN model. Doubling the data doubles the time it takes to train and predict the properties of the protein. In summary, our framework takes less than 2 hours to predict orientation and conformation. Compared to the statistical methods used by our scientists at Riken, which takes approximately a day to only predict orientation. This shows that XPSI is a promising approach towards the identification of protein structures with respect to its computational requirements. Regarding the accuracy, we present the validation error. For each test, we empirically select K for both beam intensities. In test 1, we predict orientation, and we obtain that for 99% of the data, we had an error within 2.2 degrees. For test 2, we predict orientation and conformation, and we obtain the same orientation error, and we achieve 100% of accuracy when predicting the conformation for both beam intensities. In summary, XPSI not only identifies protein structural properties in less than 2 hours, but it does it with high accuracy. For more information, reach out to us at GC Lab via our QR code.